Kun is also here, right? Kun?
Yeah.
Okay. So let's get started with a question for IRIS-EDA. So if during the break or during the yesterday, if you have any questions in using or comments, like I think you should add other functionalities to IRIS-EDA, those kind of comments, suggestions. So please feel free to ask right now. Yeah, we're gonna use several minutes to discuss those questions and the comments. Yeah, please.
During break, I tried using the IRIS-EDA website, but then somehow there has been like a proxy error in which I couldn't like see the functionality of the website itself.
Oh, okay. Do you mind sharing your slides so we can share your screen? We can maybe see that together.
Yes, I can.
Sure. Yeah, as I mentioned, for the first time you use it, will be some trouble. Yeah, but after that, gonna be okay. So Kun, can you quickly recognize what's happening there?
I never encountered this error, but I will try to see what's the cause of it. Did you just submit job on the break tab?
So you can log in the homepage of IRIS-EDA, right? And then after submitting the data, this error message pop up. Is that correct?
Yes.
Okay. So are you using the example data sets or are you using your own data sets?
I use an example data set.
That should not be the case. If using example data sets, yeah, should be. So Kun, I think previously, if the data sets format have some issue, we're gonna see such weird, error message. But if they are using example data sets, this should not happen, right?
Yeah, I just submit a break and using the example data, and it's success. And can you try again?
Okay. Yeah. Yeah, you can try again. Just submit, and yeah, start with example data sets. Just yeah. Click on the submit button. Does anything? Seems okay now.
Working right now.
Okay. Okay.
Yes.
Just like your computer, if something weird happened, just close it and reopen it, restart it.
Okay.
Computers is not as smart as human being. Yeah.
Right.
Yeah.
Thank you so much.
Okay, thanks for sharing the issue. Yeah. Other questions? So actually, we are still actively maintain and develop IRIS-EDA. So we will appreciate if there are any comments, suggestions, like what kind of functionality you prefer to use on such a server, but we currently do not have it or have them. We will try to include more functionalities in the future. Yeah. Okay. If no more questions, let me move forward. Thank you, Kun, for your support in the Rmarkdown and in the IRIS-EDA showcase. Yeah, that's really, really helpful. Yeah. Okay, so let's come back to the data analysis pitfalls. So yesterday, I introduced the 4 pitfalls. Yeah, like the first one, popularity doesn't equal to high performance. That's usually a pitfall in selecting a tool. Yeah. Recommendation is read some review paper. Yeah, and use the performance-driven recommendation. Yeah. Second pitfall is dropout issue. We mentioned this one again and again, but actually this is still a debate. Yeah. Some biologists are very against this imputation and dropout issue. They prefer to even use what they sequenced in their data, even if sparse, even only 10% of the values are non-zero values. They prefer to use what they have other than imputation, because imputation will induce and bring in a lot of false positives. They can recover those zeros into some non-zero values, but meanwhile they may, and based on the current performance, they will change those true zeros into some non-zero values, which means a lot of false positive will be brought in. Yeah. So this is still a debate. Yeah. But even without imputation, if the number of genes you called from a cell, if your target gene showcased in the non-zero values, I think that's good enough for you to answer your question. But if you want to do a kind of a discovery-driven analysis, you want to discover whether some new pathways, so new regulatory pathway or metabolic pathway are involved in this phenotype. So you may do the imputation, yeah, because whenever we do discovery-driven analysis, we should not limit ourselves by those dropouts. Yeah, so you only have 10% of genes expressed. You can never discover the other 90% things. Second pitfall is the batch effect and normalization. Yeah, compared to bulk RNA-Seq data, yeah, both bulk and single cell have a batch effect, but in single cell level, the cellular heterogeneity and other issues, including dropout issues, will make the batch effect more serious. So we should do normalization. Yeah, that is a required thing. So if you carry out your data analysis without normalization, I'm gonna guarantee there will be some problem. Yeah. The fourth pitfall is a t-test for differential expressed gene analysis. That's a kind of general statistical knowledge. Never use a t-test if you cannot make sure your data follows normal distribution. Yeah. So using some non-parametric tests like Wilcoxon test to do the differential gene expression analysis. Yeah. There are also some specifically tools designed for single-cell RNA-Seq data, yeah, which were included in my previous slide. Yeah. So that's a quick review of the first four pitfalls. Today, I still have the fifth one and the sixth one pitfall. Yeah. After these two, I'm gonna give a quick summary of the whole thing, and then I will leave quite a lot of minutes here for open discussion of anything in the past 2 weeks. I asked one postdoc and one student in my lab to further dig related review paper regarding single-cell data analysis. I think both of them have prepared some slides for our discussion. Yeah, after my summary, if we have questions or comments to be discussed in the group, yeah, we will do that first. If there's no comments or questions, my postdoc student can jump in to report their new insight from that 2 papers. Okay, that's the fifth pitfall. Yeah, co-expression correlation. Yeah, that's a very naive. I call that naive analysis. Yeah, because in R there is only one command line, like a COR. Yeah, and then you can calculate correlation. Yeah, but if you open those zoom in those parameters, there is a parameter showing you prefer to use the Pearson correlation coefficient, or you prefer using the Spearman correlation coefficient. Yeah. This is two kind of correlation calculation methods. Yeah, for example, the data, we have 2 genes and we have 10 cells. Sorry, this first one is cell 1, yeah, and there's a little bit mislabeling. But you just think about, there are 2 genes, they have expression values under 10 cells. Yeah. And then your question is whether these 2 genes are correlated, yeah, with each other. Furthermore, you can, based on the correlation, to ask whether these 2 genes are functionally related, yeah, whether they located in the same pathway, yeah, to further biological interpretation. Yeah. But when you calculate the correlation, yeah, you can use those 2 vectors and calculate the correlation. For example, the default Pearson correlation coefficient. Yeah. But here, my suggestion is in biological data, yeah, we should avoid, using the Pearson correlation. We should use Spearman. The reason is clearly showcased on my slide. Yeah. Pearson benchmarks the linear relationship, which means if these 2 genes do not have a linear correlation, then they will not have a very high Pearson correlation coefficient. But actually, based on a lot of observations and investigations, there's, rarely happen thing is the 2 genes follow a linear relationship. Yeah, most of the situation is the monotonic relationship, means they have a similar trend. They have similar patterns. That's already good enough, which means when the expression level of gene A goes up, then gene B goes up together. Yeah, when this gene A very stable and gene B is also very stable, and when gene A goes down and gene B also goes down, so that's kind of a monotonic relationship. So which can be captured by Spearman correlation coefficient. Yeah, so that's why we always recommend that you use the fundamental correlation methods as a Spearman, not Pearson. Yeah, and the good thing is if these 2 genes, follows Pearson correlation, they can also get a very high Spearman correlation score, but not vice versa. Yeah. That's the comparison, yeah, and just keep in mind when you use correlation, just select the Spearman correlation in the parameter setting. Yeah. The pitfall 6, yeah, that's the last pitfall on my list. Yeah, I know the postdoc and the students, they found other pitfalls and other common mistakes in the public domain. They will introduce later, but here is my last one. Yeah, this is very interesting. Actually, based on this observation, they have developed a computational direction, even, yeah, so-called BiClustering. I put a figure here. Yeah, in a single-cell data, as we mentioned, it's not a small big data. In the cell level, yeah, in the cell level, usually we may have an even larger number of cells than the number of genes. So the matrix is like this. A very fundamental comment is genes are not necessarily co-expressed in all the cells or cell types. Yeah. So they only co-express in a specific cell type or in a specific cell groups. Yeah, like showcased here. If you can see this red bicluster here, yeah, and this is another one. This is another one. Yeah, if you calculate correlations between 2 genes using all those expressions in all the cells, you will get nothing. Yeah, the information will be hidden in the large scale cells. Yeah, so we should observe. We should invest the data from 1 dimension to 2 dimension. Yeah, from 1-dimensional clustering technology to 2-dimensional BiClustering technology. Yeah, give this example too. Yeah, this is the same example on my previous slide. Still 2 genes and 10 cells. Yeah, you can calculate the correlation scores across Spearman correlation scores. If you calculate that, you will get minus 0.23. as the co-expression value. Then we can, based on this score, we can confidently make a conclusion. These two genes are not co-expressed at all. The correlation score is very, very low. But if you can select, you only use part of the cells, like the cells marked as highlighted as yellow, cell 1, 4, 5, 6, and 10. If you calculate the correlation score only under those 5 cells, maybe half of the cell populations, then you can get a 0.99, 0.99 correlation coefficient score. Based on this score, we can make a conclusion. These two genes are co-expressed. They are functional correlated, and here is the issue. We use the same data size, but we get different correlation scores. I personally suffered from this kind of interpretation. During our collaboration, we encountered these issues. Sometimes our collaborator generate the data, and we use 1, 2 and get the conclusion as positive, but we use another 2 generate the conclusion as negative, totally opposite directions. So even using the same 2, using parameters setting A, we generate a conclusion, but using another parameter setting, maybe it's a totally different conclusions. That's why sometimes a biologist doesn't trust in the bioinformaticians. So here is the reality of big data, especially single-cell data. That's why we recommend a model so-called BiClustering. This BiClustering algorithm was also implemented in the IRIS-EDA website. And I think there's a question in 1 to 2 p.m. regarding the QUBIC algorithm. Actually, QUBIC algorithm is developed based on this observation, several years ago. People can give a very quick solution. We just go through all the combinations of conditions and then try to find the combination of that 5 cells in yellow, and then we can capture the meaningful cell subset for these 2 genes. But computationally, that's very, very challenging. For any, like I showcased here, for any M cells, there are 2 to the power of M different combinations of cell to consider. So just imagine you have 100 cell, then the combination is 2 to the power of 100. That's a huge number. Don't even try to do that. And here, mathematical modeling can save the day. Using the BiClustering algorithm. So you can do BiClustering of the original data. Then your original data will be reorganized into another order. Reorder the gene, reorder the cells. Then you can get such a beautiful pattern here. I'm not saying every single data can be reorganized into such a pattern, clear pattern. Some data are very complicated, but this is only an example showing what is a BiCluster I'm talking about. So this is one BiCluster, and this is another one BiCluster, and this is the third BiCluster. With those BiClusters, you can identify a cell subpopulation, and along with a set of genes. Then each pair of the genes, they showcase the highly correlated pattern within this specific subpopulation. Sometimes this subpopulation corresponds to one cell type clearly, but sometimes they map to some multiple cell types simultaneously. Depends on what kind of the biological cell development. But with this BiClustering algorithm, you have a much strong power in interpreting your large-scale gene expression data among maybe tens of thousands of cells. So the underlying mathematical model is showcased here, but I'm not going into details. So basically, we are using a statistical model to give a quantitative representation of all the gene expression values in all the cells. So here is an distribution-based methodology. For example, I have gene A and 10,000 cells. I'm not using their expression values, original expression values. I will translate those original expression value into two stages. Yeah, into three stages. With the right part as a highly expressed values, and the blue part as a lowly expressed values, and something in the middle is kind of normally expressed. Then the expression vector of one gene will be translated into the quantitative, into the discretized vector into -1, 0, and 1. Yeah. So here is still this example. The same example, if you calculate the correlation among all the cells, you get -0.23. Nothing significant happened. And then we translate all the values into 1, -1, and 0. So, not considering the detail of the statistical model, you will change the larger, values into 1, the smaller values into -1, and some value in the middle as 0. Yeah, this is the output of the algorithm. You can translate the original data into a new matrix. Yeah, only consider having -1, 1, and 0. Yeah, and then you can locate those cells in which these two genes have the same expression state. Yeah, like in the first cell, both of the genes have 1, and then we think they have a similar expression pattern. Yeah, going up together. Yeah, and for the second one, -1 and 0, we will not consider it. Third cell, -1 and 1, they are totally opposite. We do not consider it right now. And then cell 4, 5, 6, both of them are going up together. And 7, 8, 9, we skip those. And then cell 10, -1, -1. So we will pick those 5 conditions under which those two genes have a same expression patterns, which means these two genes, they're going up together, going down together in this subpopulation of cells. And then if you calculate the correlation score, using their corresponding original expression values, this is 0.99. Yeah, this is what happened using this example. Yeah, and this is the underlying algorithm of QUBIC. Yeah, and after that we will build a graph model. Yeah, not going details here, but in the graph model we can identify biclusters efficiently and effectively. So here are some outcomes. Yeah, QUBIC algorithm published about actually 10 years ago. After that, it was reviewed as the best biclustering algorithm in 2012, and again, best algorithm as the single-cell data in 2019. And we even have a second version of QUBIC, and the R package and the web server. Everything regarding this algorithm, we have a kind of a whole package of this algorithm. Yeah. Actually, a lot of people use it. Citation is about 250 times. Okay. Questions? Any quick questions regarding those pitfalls? Mainly based on related to the gene expression correlation and gene module detection.
So I have a quick question about, I think, Pearson and Spearman correlation.
Absolutely.
So is there any situation in single-cell RNA-Seq when you would not want to use Spearman?
Yes. This is a very good question. The Pearson correlation can identify, they may miss some potentially correlated gene pairs, but this is a very strong signal for identify, very significant pairs. Like your purpose is not to identify all the potential correlated gene pairs. your purpose is identify those very significant pairs. Just imagine that if two genes, they are linearly correlated together with a correlation efficient score as 0.99, you can imagine how strong they are correlated, right? So in that case, you can always use Pearson to pick the top gene pairs. Yeah. But if your goal is to identify all those potentially correlated gene pairs, you should not Pearson. You should use Spearman. Yeah. And like I mentioned previously, there's doesn't hurt using Spearman. Yeah. Even you want to identify top. Because if the correlation of Pearson correlation efficient is 1, it's also very high in Spearman. It's also 1 in Spearman. You will not lose anything using Spearman. You are always identifying more. Yeah, is that clear to you?
I think so. Yeah, it sounds like when you allow nonlinearity, you're sort of casting out a wider net.
Yeah. You can identify more. Yeah, and you will not lose the linear relationship. Yeah. Because if they follow the linear relationship, their score in nonlinear relationship is also very, very high, right? Okay, good question.
That makes sense. Thanks.
Mm-hmm. Yeah, I know the BiClustering part is kind of maybe beyond the some of this knowledge modeling part. So if somebody is interested in doing BiClustering algorithm analysis and development, yeah, more than happy to talk with you offline. Okay, and we are 3:28. Yeah, let me give a quick summary. Yeah, and this is our plan. Yeah, I changed a little bit based on what we have in the past several days. Basically, we have introduction of bioinformatics and the big data. So hopefully you are not still confused at the low level. Yeah, now you should confuse at the higher level, right? Like that question: when you should you use Pearson and when should you use Spearman? Yeah, in the data analysis. Yeah. So now I do believe you should familiar with at least with the name of Seurat, right? At least with the name of IRIS-EDA. Yeah, at least the 10x genomics, SmartSeq2. Those technology names are not totally new to you. Yeah. So like we said, we introduced 2 kind of enabling technologies. Yeah, standalone R package Seurat and the web server IRIS-EDA. Yeah, they are only representatives for the 2 kind of techniques. You can easily find other R package, Python package in the public domain. Yeah, roughly there are 680 something single-cell RNA-Seq analysis tools. I mean those standalone tools in public domain. Yeah, all the tools are summarized in the link I shared on the second day. And there are also a lot of web servers, web databases in support of the single-cell RNA-Seq data analysis. You can always get started with Seurat and IRIS-EDA and explore more based on your question. And we introduced the single-cell RNA-Seq basic analysis pipeline and philosophy, following with several pitfalls. Yeah. Like I said, instead of giving a list of do's, I further prefer give you some do nots. Yeah, so you can, because for the do's, you can just select some Seurat package. They already designed a very comprehensive functionalities. But just remember, there are some pitfalls in using those tools in analyzing your data. Yeah. And then today we finished all the pitfalls and gave the summary here. And here is the acknowledgement page. First of all, I will send a special thank you to Jeff, Rusty, and Naren Without your support and the coordination, we cannot have this 2-week class. Thank you very much. And I also want to send my thank you note to my lab members, especially Yuzhou Chang and Cankun Wang. Yuzhou is the person who delivered all the information for Seurat, and Cankun is the person who delivered Rmarkdown for developing pipeline and the IRIS-EDA web server. And we still have other students contributing this class. Zhenyu Wu and Yang Li, they have prepared some slides. If we have time, they will discuss more regarding single-cell RNA-Seq data. And Anjun is the senior PhD student. He was here for some questions, for some dropout issue pitfalls. And thank you all for the people who contribute. And I also want to thank the NIH grants. First of all, is this current R25 supporting the whole big data project at ISU. And I also want to acknowledge my R1 project, focusing on single-cell data analysis and modeling. Some of the tools like IRIS-EDA and the QUBIC, they are supported by my R1 project. Okay. So Nastia and Naren, I want to stop here. I think that's all my part. And let's see what kind of question or comments here we can discuss together.
Sure. Thank you so much for the kind words, and we really appreciate your time and the way you deliver the lectures. It was very easy, productive, and I hope we all learned a lot. Especially in my case, I learned a lot from you. And I must say, you have a very good teaching skills. So I thank you so much for sharing your knowledge. Thank you so much for going through each and every step so that we understand it correctly. And now the forum is open for the students. So undergrads or faculty, if you have any questions, so please, we can discuss that. If we don't have, then we can wind up this session. Any question, any comments?
So we can ask about anything right now?
Yeah, so please.
Okay. So I don't want to catch you guys off guard, but you mentioned an imputation project where you guys were doing this kind of self-converging thing. I'd be really curious to know more about that.
Yeah. So, like I said, even it's a very big debate in the public domain regarding imputation, but including our lab, a lot of reputed labs and groups are developing imputation methods using the deep learning framework, because the autoencoder framework in deep learning is a natural framework to do imputation. Yeah. So first of all, we're gonna do embedding, which means we're gonna select and extract the important features from the original data, and using the deep neural networks, and we will recover those gene expression values based on the embedding features. So this is the power to recover expression values who was there originally. Yeah. A lot of tools in public domain are doing this, but they are not controlling the false positive very well. So like Anjun showcased, there is a tool named ISU in our lab. We are currently submitting a paper. Actually, this is an invited paper by Nature Method. Editor of Nature Methods, they're very interesting in our work, and we are submitting this one to the Nature Methods. But during the way we're preparing the manuscript, we found that the space there is huge. Yeah, there's a huge space in developing new technology, in interpreting different kind of single-cell data. So we do have some new ideas in developing new tools or enhance our current tools. So yeah. Feel free to contact me offline if you or anyone else are interested in this imputation direction. Okay.
Okay. Cool.
Yeah. So like I mentioned, Yang Li, who is my postdoc, he sent me this paper. Yeah, he sent me this paper last week, and I quickly found some very interesting things we can discuss together. The title is like 10 common statistical mistakes to watch out for when writing or reviewing a manuscript. Yeah. So I personally like those. At least we can review the 10 common mistake list, right? Yang, you can jump in, and I'm changing this slide to you. You can give us a quick introduction for discussion. Okay.
Okay. Good afternoon, everyone. I am Yang Li, postdoc in Dr. Ma's lab. Then I will discuss several common statistical mistakes when writing or reviewing a manuscript. This summary comes from a paper whose titles and authors is pasted on the previous page. In this paper, 10 common mistakes are summarized as below. Today, we won't go into details for each of the 10 mistakes. Here, I will select 2 typical mistakes to discuss. Please switch to the next slide.
So before going into details, I'm gonna quickly discuss the list. I know some kind of terminology are very computational, but there are some very the third one, interpreting comparison between 2 effects without directly comparing them, and using small samples, circular analysis, feasibility analysis. I think those are very interesting things. Like the last one, correlation versus causality, causation. So that's another very interesting topic. Like in statistics, people always say, "Hey, we are looking for real causality," but 99% of the causality-driven analysis are using correlation, not real causality. Okay, Yang, you can go into details regarding the first one. This one.
Yeah. Okay. And the most common mistake is failure to correct for multiple comparisons. And to illustrate this mistake, I use pathway enrichment analysis as an example. In pathway enrichment analysis, our gene set is compared to each of the pathways in database. Each comparison will generate a p-value. However, it is inappropriate to directly use this p-value as a significance cutoff. Because multiple comparisons are needed for multiple pathways. And the next slide. Thank you. Specifically, comparison to a single pathway at a significance level, alpha equals 0.05 means the probability of observing that the gene set is enrichment in the pathway due to chance is no larger than 0.05. However, if there are 20 pathways to compare, the probability of observing that the gene set is enriched in at least one pathway due to chance is 0.64. That means we have a 64% chance of observing at least one pathway that the gene set is enriched in, even if each of the tests are not significant. And the solution to this problem is to correct for multiple comparisons. For example, the Bioinformatics correction. Bioinformatics correction is performed by dividing the number of comparisons by the significance level. In this example, the error rate, the real error rate after correction is 0.0025, and the formula to get this result is written on this page. This is the first mistake.
Yeah, quick summary of this Yang's problem. So basically, this is very similar to the p-value adjustment I mentioned early today. The thing is, in one word, yeah, if you pick the first one, you only pick top one, nothing to be worried, nothing needed. Yeah, just use the original p-value, pick the top one. But whenever you select multiple candidates, keep in mind we have to do p-value adjustment. We have to correct our p-values. Yeah, which means we need to do the multiple comparison. Yeah, the reason is very simple. Yeah, when you select more candidates, the probability of making a mistake is high. Then you need to adjust your p-value. Yeah, so basically this is the same thing Yang just showcased. Yeah. Okay. Do you have other? Okay, second one. Yeah.
Okay. The second common mistake is spurious correlations. We use an example to illustrate this mistake. To calculate Pearson correlation between two variables of 20 points. And firstly, look at figure ABC. We select one point as outlier, that is the red point, and move it away from the other 19 points. With the increase of the distance between the outlier and the other points, the Pearson correlation coefficient becomes bigger and bigger. However, there's no significant correlation between them.
Mm-hmm.
And then look at figure D, E, F. We partition the 20 points into two groups and move them away from each other. With the increase of distance between the two groups, the Pearson correlation coefficient also becomes bigger and bigger. However, in these two cases, there is no significant correlation between the two variables, and this is called the spurious correlation.
Yeah, that's interesting. Yeah, it's a kind of extended discussion of my previous slide, Pearson. So Pearson versus Spearman. Even you use Pearson, there are some fake, correlation score. You observe a high correlation score, but actually they are not real high. Yeah, I think this is a point. Okay. Okay.
Please switch to the next.
Mm-hmm.
The usual solution to this problem is to replace Pearson correlation with bootstrapping correlation. And the calculation process of bootstrapping correlation is as follows. First, randomly sample n points from the total set and calculate the Pearson correlation coefficient R star among these selected points. The n points compose a bootstrap sample. And then we repeat the sampling process for m times and calculate the bootstrapping correlation by averaging correlations of the m bootstrap samples, which is written on which is presented in the formula on this page.
Mm-hmm. Actually, Yang, this comment is to you. Actually, this is a statistical formulation of our backcasting algorithm. Backcasting is a kind of combinatorial algorithm. So you just get started from some very good seed and grow them to a backcluster. Actually, the underlying idea is very, very similar here. You just bootstrap, which means you select the set of cells, and then you calculate the correlation and take a mean. Yeah, and okay, yeah, that's interesting. Do you have more?
No, that's all.
Okay. Okay. Appreciate. Yeah, there's some very interesting discussion here. I think we can still have several minutes for Zhenyu. Yeah, this is a new PhD student in my lab. he sent me this paper, 11 ground challenges in single-cell data science, which was published, I think, just in this year. So, Zhenyu, you have some very interesting insight to share here, but let's keep in 5 minutes, okay?
Okay. Yeah. So, so in this paper, the authors present 11 grand challenges in the single-cell data analysis. Please go into the next slide. Yeah, some of the challenges are already mentioned in our previous lectures, like the trajectory inference and the sparsity. So in the next 5 minutes, I will mainly focus on the sparsity handling and then the spatial pattern finding and the integration of the single-cell data. So
okay,
the job.
Zhenyu, considering we already discussed the sparsity, can we just mention spatial analysis and integration analysis? How about that?
Okay. Yeah.
Oh. Yep.
Yeah. So let's take a quick look at the previous single-cell isolation technology. So whatever you are perform the dropseq using the droplet and the microfluidic tag, or you choose to use the cell sorting single-cell isolation technique. So all the spatial information are lost during the isolation.
Mm-hmm.
So how to preserve that spatial information during the single-cell RNA sequencing is a challenge for us to find the spatial patterns. So one solution is to perform the single-cell RNA sequencing in situ. Please go to the next slide. Yeah. Sure. The 10-time genomics offer us a commercial way to perform the spatial RNA sequencing. So basically, just they combine the so before you perform the single-cell RNA sequencing, you first do the HE staining. Please click. There's animations in this slide. Yeah. So you first perform the HE staining in your sample, and then you did. There are a lot of tiny wells beneath the sample, and those the single cells or several, like 2 or 3 cells, will fall into the tiny wells beneath the sample, and then you can perform the single-cell RNA sequencing in situ, and thus all the spatial information are preserved. So that's one solution which solved by sequencing technology. And another solution is the single molecule fluorescence in situ hybridization. So in 2014, the Luca had developed the single molecule fluorescence in situ hybridization technique. So basically, they use a probe to detect the expression of certain mRNA, and they work the probe out, and they use the same probe, but with carrying different fluorescence mark labels. And after several cycles, they can detect the precise RNA expression in certain cell or in certain tissue. And in 2019, they have modified this technique, so which allow us to use only one single probe to detect one mRNA. Please click. Please go into the next slide. Yeah, this new probe, which carrying 4 different fluorescent markers, allow us to detect one mRNA at the same without washing out and rebinding. And please note that all the experiments are carried out in situ. That means all the spatial information are preserved, and then next picture shows the result of the single molecule fish, and yeah. So that's the second way to preserve the spatial information. Yeah. And the last challenge I was mentioning in this slide is the integration of the single cell data. In this figure, you see there are already like 7 to 8 methods allow us to combine the RNA sequencing data with other high throughput sequencing results, like the Hi-C or ATAC or the DNA methylation. But the combination of the RNA sequencing data and the other sequencing data like the ATAC or Hi-C could help us to validate the accuracy or the precisity of the single cell RNA sequencing data. And please go into the next slide.
Yeah. So before that, I have a quick comment. Actually, this paper is published in my lab. The first author was Anjun. Besides the single cell RNA-Seq data, now currently a lot of technology can generate the match the single cell RNA-Seq and other modality simultaneously from the same cell population. Yeah. Most of them are not commercialized so far, mainly developed by single labs. But in the future, we are expecting to have a lot of matched, single modality data in a single cell level, multi-modality data in a single cell level. In one cell, we can get their RNA-Seq, get ATAC data, mutation, yeah, maybe spatial information simultaneously. So that's another very promising field if some students want to go into single cell data analysis. Yeah. Okay, this is for you.
Okay. Currently, we have 10 methods which allow us to analyze to combine the multi-omic single cell data and to do the cross validation and give us some results. For example, the Seurat 3, the most famous one, it can co-embed the single cell RNA sequencing data with the unmatched single cell ATAC data. And we can use the combination to predict the cell type specific reticulum. Besides, the Seurat also can apply the spatial transcriptomic data to predict the spatial gene expression patterns. Besides, I want to mention it's the DECO. The DECO can analyze the gene expression with the cell the neighboring cells, and allow us to construct a unique spatial network. And it's the only one of few tools that provide us with a web-based interactive visualization and explore. Yeah.
Okay. So Zhenyu was mentioning the last three tool, so-called GIOTTO, GIOTTO, developed for the imaging data analysis together with the spatial information, spatial single cell RNA-Seq data. Yeah. So this is a very developed tool, have not been published in any journal, but they have a bioarchive paper, and the tool is currently available to use. Yeah. Okay. Good. Do you have other things to share?
No.
Okay. That's good enough. Yeah. Thank you, Yang. Thank you, Zhenyu, for digging out the additional 2 papers. Yeah, I personally learned something from these 2 papers. Yeah. Okay, and we are almost done. And maybe one more questions or one more comments to discuss together. Yeah, this is a lab information showcase here again, showing what we do. Yeah, and main research interest. All the informations can be also accessed from the link in the bottom of the slide. More than happy to work with faculties, GRA, and undergraduate students here in any way you prefer. And that's it.
Just a quick question.
Sure.
So the second point, regulatory DNA motifs prediction. So are you using some kind of tools to predict the motifs and then verify through the wet lab? Or are you going to find out some enhancers like cis or trans regulatory modules to see how they work? So it would be fine if you just give some insight on this particular topic.
Yeah, thank you for the question. Actually, the motif analysis direction is also long-term interest within the lab. Actually, I get started work on this topic in my PhD and postdoc period. We focus on algorithm design. Yeah, we focus on algorithm design and enabling tool development. We hope we can find collaborators in validating our TF motif binding site predictions. Yeah, so we should have new algorithms, tools, and web servers in support of DNA motif prediction, actually including some RNA motif prediction. Yeah. So basically, this is not related to single cell so far, but the main data type we worked on, including the promoter enhancer regions directly from the genome, or the ChIP-seq data from some experimental sequencing. And recently, we have ATAC data. Yeah, using ATAC data, we can also improve the DNA motif predictions. So basically, it's data-driven, the algorithm design and the tool development. That's what we do in the lab. Yeah.
Okay, thank you.
Yeah, but we are very eager to find the collaborators who can work on this direction, because the motif prediction and validation could support this gene regulatory mechanism in an organism, in a genome model, or in a specific cell types. Yeah, very interesting and very important topic. Yeah. Okay. And Run, I will share the materials today with you with a link as previously, and along with the email. Okay.
Okay, thank you. And I will send you the videos. Is it okay if I send it later today, like around 7 or 8?
Absolutely.
Okay.
Yeah, absolutely. Yeah.
Okay, thank you. Thank you so much.
Last minute, thank you all in this class, and I really enjoyed working with you all. Yeah. Okay.
Okay, thank you so much. And any quick comments, anything? Students? Okay.
Thank you for this presentation.
Oh, thank you. Thank you for your good work.
Thank you.
So hopefully everyone have a good rest of the day and keep healthy. Yeah.
Thanks
so much.
Yeah. Bye-bye.
Thank you so much. Take care.